sort of anyway. Okay, so we mentioned two neuroglial cells in the peripheral nervous system, right? What were the two? Schwann cells, okay, and satellite cells. Satellite cells, we're not really, no one's really 100% sure what all they might do. They think that they do what, though, or have the ability to, absorb neurotransmitter, good. Myelin sheaths, however, in the peripheral nervous system are made up of the Schwann cells. And what's myelin do? Insulates the axons and allows the impulse to travel faster. Okay, cool. So when we hear about gray matter and white matter's talking about myelinated axons, right? Okay. So myelin sheath, did we hit this slide yet? No? Cool. Okay. I sort of got to put a new battery in this thing. It's just like pulling teeth. All right. So, it's an insulating layer around the nerve fiber. Let's try this one. Look at that. And in the peripheral nervous system, this is done by the Schwann cell. These Schwann cells are primarily fat. Okay? And this is what's going to insulate the axon. This is what's going to speed up conduction. This is one of the things that's still developing in like young, very small children under the age of 3, 4. Their nervous system's still developing. Part of it is myelinating these axons. If you've noticed, little kids don't have very quick reflexes, especially little like babies. You can sit a little baby up and take a little softball and toss it to them, and you watch it bounce off their head and roll back to you, and then they go like this. Okay? That's because that whole circuit of seeing it, telling their hands to try to grab it, are a little slow. Okay? And it's because these axons aren't always myelinated yet. And it's also why you don't want to put an infant or a small child on a no-fat diet. You know, mom might do that because mom's thinking, I'm going to lose this baby weight, I'm going to make this kid, it's going to be the healthiest kid ever, we're going to eat no fats and whatever. Well, their nervous system isn't going to develop properly. All right? In between each myelin sheath, now we saw an axon, we saw the myelin sheaths, and there's always a little space between them. And that's called a node of Ranvier. So for defining them right now, we're just saying they're the gaps between the myelin segments. These gaps have lots of voltage-gated sodium channels. Okay. We had another area on the neuron that had a large aggregation or large number of voltage-gated sodium channels. And what was that area called? The axon hillock. Okay. We can also call it the trigger zone. Okay. So the trigger zone is technically this transition area between the soma and the initial segment of the axon. Okay. I kind of lump them together. Axon, helix, trigger zone, I make them the same thing just to make it simpler. And this is where our action potential, this is where our impulse that's going to be conducted down the axon actually starts, is in this trigger zone of this axon hillock. Okay. So we got a pathology here. MS stands for what disease? Multiple sclerosis, okay? And this is actually a breakdown of the myelin sheaths, okay? So in their nervous system, the myelin sheaths are starting to be broken down, and it's going to be replaced by scar tissue, but essentially, we don't insulate that axon anymore, so now it doesn't conduct as quickly as it used to. So the speed of conduction itself is disrupted, and we're going to have symptoms depending on where in the nervous system this takes place. Okay? But you can imagine that if this is in motor neurons going to skeletal muscle, their reflexes and/or their ability to control skeletal muscle is going to be much more difficult for them to do. Okay? If it's going to their legs, their balance is going to be thrown off. Because if you're walking and you stumble, if your neurons are everything's firing fast enough, you'll put a hand up, you'll balance, catch yourself on a wall, you'll kick a foot out, brace yourself. If your axons aren't myelinated and you can't do that quick enough, you can't react, and so when they stumble, they're going down. Okay, does that make sense, what I'm saying there? So it's almost like they go back, like time reverses itself, and now they're going back almost to being like an infant type of thing. Okay. So here's kind of what that myelin sheath looks like. It's a Schwann cell, and it's like taking a tube of toothpaste and starting at the bottom and coiling it all around the axon until the nucleus and the bulk of the cell cytoplasm are pushed to the top. And they'll refer to that then as the neural lemma. And the neural lemma is going to come into play here and be important because we're going to need the neural lemma to be there in order for axons in the peripheral nervous system to regenerate themselves. Here's a space between two myelin sheaths, so that would be a what? Node of Ranvier, and have a high number of, voltage-gated sodium channels. Good. Okay. So nerve single speed depends on two factors. One, the diameter of the fiber. The greater the diameter, the faster it will conduct, and whether or not it's myelinated. If it's myelinated, it's going to be faster than unmyelinated, as long as the diameter is the same. And so they have three types of fibers. They call them type A, type B, and type C. A type A fiber has a large diameter and is myelinated and will conduct impulses at about 120 meters per second. Okay? So that's from one end of a football field to the other, with the end zone probably factored in there as well, in 1 second. I think it equates to about 300 miles per hour. So that's really fast, considering how big we are and how the distance an impulse might have to travel. It's going to get there really, really quickly. Small myelinated, so now we decrease the diameter. We still myelinate it. Now it's 3 to 15 meters per second, or what equates to about 40 miles per hour. Okay? Still, 40 miles per hour from my brain to my big toe is really fast. Okay? And then the last are small unmyelinated fibers. So now we get even smaller diameter and we no longer myelinate them, and now they're 1/2 to 2 meters per second, or what's about 2 miles per hour. Okay? So still has a speed to it, but not nearly what 300 miles per hour is. Okay? And these are the types of fibers that you can find in your body. All right? Obviously, anything that has to deal with keeping you alive is going to be on a large myelinated fiber. Okay? Usually things on the inside of the body that we need to get information from, but speed probably isn't that imperative, they'll be on type C fibers. Okay? They can't be all type A fibers. If all the neurons in just your spinal cord were type A, were all myelinated, your spinal cord would be the size of a trash can. Okay? You wouldn't look right. People would not, you would not have offspring, because nobody would want to be with you. Okay? You can still find those in the brain and kind of transition areas between the brain and the body. To give you an idea on speeds, this won't do much for females other than what you've seen from the outside world. Guys will totally get this. So your testes used to be up inside your abdominal cavity, up by your kidneys. Before you're born, they usually descend down into the scrotum. So the skin of the scrotum is innervated by type A fibers. Your testes are actually these type C fibers. So guys that ever get hit in the stones, you can attest to this. There's this, you get hit, and you're like, ooh, that hurt. And then it's like 1001, 1002, and then this wave of pain and nausea like you've never felt before hits you. That's the difference. That's the delay. All right? And ladies, you might have noticed that you accidentally graze somebody there, and you watch. Oh yeah, it starts to kind of get nauseous and sick because you got him. You always act like it's on accident. Oh, I didn't mean to. Yeah, whatever. Giggling about, oh, did it hurt that bad? Okay. What else? Another, I guess, analogy would be type C fibers do pupils. So if you go from dark to light or light to dark, that time it takes your eyes to adjust, that's that slow impulse conduction for those. Function. Slow signal supply stomach, dilate the pupil. Those would be type C fibers. Type A, innervate our skeletal muscles, sensory signals for vision and balance, things to keep us upright, able to see a predator, fight off the predator, run away from the predator. Those all have to be on type A fibers to give us a fighting chance. Okay. Regeneration of a neuron in the peripheral nervous system. So, in order for this to happen, we have to have the soma of the neuron still intact. That's the cell body, the cell brain. You kill the brain, the whole neuron dies. That's why spinal cord injuries are a problem, because that's often where these cell bodies are located, and then the axon comes out from there. And then you have to have the neurolemma intact, which means you have to have one of the Schwann cells still intact. Okay? So some soma and some neurolemma must be intact. Because we only have the neurolemma with Schwann cells, we only have this ability for neurons to regenerate in the peripheral nervous system, not the central nervous system. Okay, that's why, once again, a spinal cord injury, when somebody's paralyzed, their legs, paraplegic, quadriplegic, they don't get those back. Okay, they'd like to. They want to start to try to regenerate stem cells into neurons, but at this point, can't do anything about it. So what happens is, it's really pretty simple. So we're showing a neuron here, multipolar neuron, axon going down this way, looks like it's innervating skeletal muscle. We get some sort of damage here, okay? So we're going to have the proximal end of the neuron, we're going to have the distal end of the neuron. The distal portion, the portion furthest from the soma, the cell body, is going to degenerate, okay? This portion of the neuron is going to start to send out what they call sprouts. Little neurons trying to find their way back to where they need to go. But they grow very slowly, and they have no idea where they're going. Okay? And what actually happens is that neurolemma grows a tube. And the tube, think of it almost like PVC pipe, goes back down to where it used to synapse. Okay? And so then what you need to have happen is one of the sprouts needs to make it into that PVC pipe. Once it's in there, that pipe guides it to where it needs to go. And it also produces what are called neuron growth factors, which makes it grow faster and stronger. And once that happens, when one of the sprouts gets exposed to the neuron growth factors, the other sprouts just kind of degenerate. Okay? So we don't have actually neurons innervating things that they shouldn't. And then it will reestablish, it'll grow down the PVC pipe, it'll reestablish its synaptic contact, and then everything should go back to normal. So in that setup, you can see that there's a possibility neurons will regenerate, but conceivably too, if none of those sprouts make it into the neurolemma, the tube that the neurolemma made, then you might not establish the connection again either. So you just have a chance of it. It doesn't mean that it definitely will. So the stranded end of the axon and the myelin sheaths degenerate. A regeneration tube is formed by the neurolemma, one of the Schwann cells that was intact, and follows the path of the original axon. We hope that one of the sprouts makes it into that regeneration tube. If it does, that tube guides it down to where it was, where it had been, and also secretes neuron growth factors onto that sprout to help it grow faster and stronger. At that point, that causes the other sprouts to retract. Okay? They stop growing anywhere. And we reestablish the synaptic contact we once had. Okay? So that seems pretty easy, I hope. So we got our first question. No, we don't have our first question. I thought we did. So here's just a picture trying to show us this. We damage the axon. Okay? Macrophages come in and they clean up this area. All right? We make a regeneration tube. We hope that one of the sprouts finds its way into the regeneration tube where we expose it to neuron growth factors. If that happens, this will grow all the way down to the end, reestablish synaptic contact, and everything will work the way it was. So, which of the following needs to be present in order for a neuron to regenerate? I like 4 too. 4 is the answer. Okay, we need the soma and the neurolemma. That's all we need. Oh, electrical potentials and currents. I think we hit this in the last unit. Okay, so this should be somewhat of a review. So we know we have the neurons. Neurons synapse on other neurons. It's generally not a direct one neuron from the brain all the way down the spinal cord, all the way out to its effector. Okay? It's a neuron maybe from the brain, down the spinal cord, synapses on another neuron that goes out to the effector. Generally, there's at least 2. Okay, so nerve pathways are not continuous. They're a series of separate cells synapsing on each other, one telling the next one what to do, telling the next one what to do. And these connections are based on those electrical potentials, which I think is, we touched on this. This is where it should be reviewed for what we saw in the last unit. It's the difference in ion concentrations across the cell membrane. Okay, so we know we have lots of sodium outside the cell, lots of potassium inside the cell. There were large negatively charged substances. What were they? Remember the P? Not potassium. Large negatively charged anions inside the cell. Protein? We talk about that protein? Sometimes it's like Groundhog Day for me, so we may not have. You may be like, dude, I have no idea what you're talking about. Okay, there we go. All right, so we're going to have our membrane, okay? We're going to say this is the inside of the cell, this is the outside. The outside has lots of sodium. The inside has lots of potassium. It also has large substances in there that are negative charge. Proteins. We don't have to put this down here. We'll just say outside and inside. Okay. Because of these negatively charged proteins, the inside is going to have a negative charge. We have the sodium-potassium pump that pumps out 3 sodiums for every 2 potassiums it pumps in. Okay, and those are going on all the time. So even though I'm only drawing 1, I just want you to know lots of sodium out here, lots of potassium out here. We have the leakage channels. Okay, does that sound familiar? Okay, so potassium's allowed to move. Sodium doesn't get to as much. Potassium gets to go where it wants to go. So diffusional force inside pushes potassium out. Electromagnetic force of positive wanting to go to the negative inside draws it back in, and at some point it comes into equilibrium. Does that sound familiar? Okay, and that sets up our resting membrane potential. Neurons have resting membrane potentials just like skeletal muscle do. Okay, so the electrical current is the flow of these charged particles from one area to the next, and it's going to be the flow when we open up channels. Okay, and I'm pretty sure we talked about this as well. Sodium on the outside, lots of sodium, diffusion is going to push it inside, right? Also, sodium is going to be drawn to the negatively charged inside. So both forces, diffusional and electromagnetic, are going to pull sodium to the inside, right? This comes in, we're going to end up making the inside positive. Okay? So think of this as part of a axon. So in here is going to become positive, but here's still negative, here's still negative, here's still negative. Sodium rushes in, makes it positive. Now, potassium, that was being held here by that negative charge, diffusion is going to push it out. So we kind of get this reversal. And then this positive charge is going to open up more voltage-gated sodium channels, and sodium is going to rush in again and make this area positive. That's going to open up more voltage-gated channels. Sodium rushes in, makes this area positive, and that's what gets conducted down the length of the axon. Okay, is this flow of sodium in and potassium out, and then the sodium-potassium pumps reset that. They pump the extra sodium back out and potassium back in. Okay? Because of this, we say living cells are polarized, meaning they have some sort of charge. All right? The resting membrane potential of most neurons, depending on what book you're looking at, is about negative 70 millivolts. A flashlight battery is about 1,500 millivolts, so it gives you an idea. It's not a very strong charge. Okay? But if you go to Sedona and those pay the money to go get like the vortex or whatever, that's supposed to be helping with some of those charges of your cells. I don't know. I've not done it. I have no idea if there's any merit to that or not. Seems a little off the wall, but what the hell. Okay. So, resting membrane potential, we have this unequal electrolyte distribution. So that's just a fancy way of saying we have lots of sodium outside the cell, lots of potassium inside the cell. Unequal. Okay. Sodium outside is not equal to the sodium that's on the inside. These are our electrolytes, okay, between the extracellular fluid and the cytoplasm. And it's because of the stuff we already mentioned. We have large cytoplasmic anions, proteins being some of those things, phosphates, sulfates. They all carry a negative charge. They're trapped in here because they're too big. So no matter what channels open up in the membrane, they're too large to exit. So because they're stuck in here, it tends to make the inside more negative. We've got our excitability pumps, the sodium-potassium pumps, working all the time, pumping 3 sodium out for every 2 potassium it pumps in at the cost of 1 ATP. Okay. So just to set up this resting membrane potential, the body's using energy all the time. Okay. The ATP, the energy is used to set this thing up. Okay. Then when the channel's open, diffusion takes over. Okay. Diffusion and electromagnetic pull. All right. So it's that story. It's setting up that scenario. It's setting up that potential energy. Okay. It's Wile E. Coyote pushing the boulder to the top of the cliff to try to get the roadrunner. Okay. Membrane's very permeable to potassium. It diffuses out because diffusion, high concentration to low concentration. It doesn't like being crowded in here. So it starts to move out. As it moves out, it tends to make the inside even more negative, which then pulls harder on it for it to come back in. And at some point, the amount leaving equals the amount coming back in, and we call that our equilibrium. Okay? The membrane is much less permeable to sodium. Sodium's pretty much stuck out there until we open the doors to let it back in. Any questions on that? I know it's kind of review, but maybe some people didn't really get it the first time, and they're not getting it here now the second time. Maybe we didn't care the first time, and we still don't care the second time. I don't know. All right, so here's a picture supposed to be kind of showing us. This is supposed to represent a phospholipid bilayer, which is a membrane, which is a cell membrane anyway. Here's our channels. The purple dots, purple balls are sodium. You see lots more purple out here than in here. You still see some, but it's more concentrated sodium out here than in here. The yellow is supposed to be potassium. You see potassium outside the cell, but there's lots more inside the cell. And then these large anions, might be the phosphates, it might be the proteins, whatever. They're too big to leave. Okay? So you can see that just on diffusion, if we just look at the yellow dots in here, they would want to move out here and even out the number of yellow on both sides, right? That's what diffusion would want to do. But because of the negatively charged inside of the cell, they're drawn to that. So for each one that leaves, there's one being drawn back in, and at some point that evens out. Okay? Sodium doesn't get that ability to do much. It's kind of stuck out here. But when we open the doors, it comes rushing in. Okay? Let's see if we got a video here. Probably one we've already seen. Oh, we're not going to be able to hear it because of this microphone thing. Well, let's see we can just see if we can watch it here. Inside cell, okay. So we got the anions, the potassium stuck on the inside. Their concentration gradient diffusion wants to push it out this way. He's still talking. We don't know what he's saying. Come on, move it along, move it along. So potassium is allowed to leak out a little bit because of that diffusional force. Come on, come on, come on, come on. But because of the negative charge inside, that's going to be the electrical force. It's going to pull some of it back. And pretty soon the amount leaving equals the amount exiting, and that's our resting membrane potential of negative 70 millivolts. There they are, coming into equilibrium. Exciting stuff. All right, well, that was a waste. So sodium concentrated on the outside, potassium concentrated on the inside. Hopefully with the pictures we've had, you have a good visual, a good understanding of what this resting membrane potential is. So the outside of the cell is what? What charge versus the inside? Positive. So we normally look at is the inside of the cell is more negative. Well, the outside is more positive. So I just switched it up a little bit to see if you'd still get it. And there was no tricking you anymore. All right. So local potentials. We dealt with local potentials before. That was in our neuromuscular junction. That was the neurotransmitter coming across the synaptic cleft and opening chemically gated channels for sodium. Okay. If we let enough sodium in, we reached what? What was that thing called? Not equilibrium. There with a T. There with a TH. Local potential, if we let enough of the sodium in with the neurotransmitter, we reached something which led to the opening of threshold, which led to the opening of the voltage-gated channels. Okay, that neurotransmitter letting sodium in was our local potential. Okay, so they're local disturbances in the membrane potential. They generally happen on the soma or the dendrites. Okay, from another nerve synapsing on it. Okay, so they occur when the neuron is stimulated. It's opening chemically gated channels. And this leads to sodium being able to come inside. We call that depolarization, right? So we have the depolarization from chemically gated sodium channels. Sodium rushes in and makes the inside more positive. But there's not that many of these chemically gated channels. So there's a limited amount of sodium that gets to come across. So there's a limited amount of charge change. But if we change the charge enough, that's the stimulus to open up the voltage-gated sodium channels, of which we said there's lots of them located where? Axon helix or the trigger zone. Okay? And when those open up, they let lots of sodium rush in, and that's what starts our action potential. So local potentials can lead to action potentials. They don't have to, but they can. Okay? They have to hit threshold in order to do that. So local potentials are graded, meaning not every local potential is the same strength as another. It has to do with how much neurotransmitter gets released and for how long it keeps getting released. The more neurotransmitter that comes across, the more doors we open. The longer it comes across, the longer we leave the doors open. Each of those equates directly to how much sodium gets to come inside, right? The more sodium that comes inside, the more we change the charge inside. Following that? Okay. Somebody nod, just to make me feel better. Okay. Local potentials are decremental. They're weaker the further they spread. Okay. So sodium runs in, or a bunch of sodium runs in, and it starts to move its way down membrane, one direction or another. The longer it's in here, the further it goes, the more likely the sodium-potassium pump's going to go ahead and pump some of that back out. So if sodium rushed in, let's say 3 sodium rushed in, and they started to move this way to make the inside more positive, but the sodium-potassium pump grabs them and pumps them out, we lost that change in charge, right? Okay. So the further it has to go, the weaker it'll become. It's reversible. Okay. So it's going to go back to this picture. Okay. We allow sodium in. Sodium brings a what charge with it? Positive. So as it comes in, more sodium comes in, it's going to make the inside more positive, right? Less negative. And this usually happens to a point, and then the potassium channel's open. Okay? So it's sodium's rushing in, and then potassium channel's open. If sodium's rushing in and at the same time we're letting potassium escape, are we going to gain much? No. Okay? Sodium's coming in with its positive charge, but potassium's leaving with its positive charge. So we get no net charge change. So that's what it's saying, it's reversible. If we do something to open these potassium channels early, it kind of negates the sodium rushing in. Okay? Local potentials can be either excitatory or inhibitory. Now, we've only looked at channels, chemically gated channels for sodium. Okay? Chlorine is another ion or electrolyte that's oftentimes concentrated outside of the cell. And we have channels sometimes for that. And we can open up channels and let chlorine come in. Chlorine has a negative charge. If we let chlorine come inside, are we making the inside more positive? No, we're making it more negative. So now this is going to be less likely to fire because the resting membrane potential in here, where'd my marker go, is minus 70. To actually get the threshold and hit our action potential, we have to get it to minus 55. Okay? So if I take chlorine in, we're making minus 70. Maybe the inside's not going to be negative 70 more. Chlorine comes in, maybe it's going to be minus 72, maybe 74, minus 75. We're getting further and further away from minus 55. So if we did at the same time, we let chlorine in and then we let sodium in, we'd need more and more sodium than we normally would to make this change, right? All right. Okay, so those are local potentials. Generally, for what we're looking at, they always revolve around the neurotransmitter, the chemically gated channels. Okay? So if you see something about chemically gated channels, at least for us, you're thinking local potential. Okay? Which if it hits threshold, then we change the voltage enough. This is now the stimulus that opens up voltage-gated channels. And when you think of voltage-gated channels, now you're thinking the action potential. Local potentials take place on the dendrites and the soma. Action potential takes place going down the axon to the synaptic knobs. Okay. So here's a bipolar neuron. We're stimulating these dendrites or this soma somehow. Another neuron synapsing on it, or multiple neurons are synapsing on it. And we get these local potentials. If it gets enough stimulation and the local potential is great enough, we hit threshold. Threshold causes all the voltage-gated channels to open, of which we have a high concentration right there in the trigger zone, or the axon helic. That allows lots of sodium to rush in, move down. That rush of sodium opens more voltage-gated channels here on the membrane. It rushes in, opens more voltage-gated channels here, and it's like a domino effect all the way down till it goes to each end of this synaptic knob. Okay? Are we following me at all? Okay, cool. That's the action potential, yeah. So this is all over here, local potential. From here on down, that's our action potential. Okay? And these local potentials may or may not lead to that action potential. This stimulus might not be enough to get an action potential. Okay? Sometimes you need a greater and greater stimulus to get a reaction. Those of you that have kids, can you always just get away with nicely asking them to pick up their room? No, sometimes you got to open up a can of whoop ass on them. All right? That's a stronger local potential. When you step into the room with a belt or something you're going to beat them with, then they start to like, holy shit, Mom's serious. We're going to start picking this thing up. Okay? So, action potentials are diagrammed. They're usually referred to like a spike. The characteristics of now an action potential. So we just had 4 characteristics of a local potential. They were graded. They can be inhibitory. They can be reversible. And I'm already forgetting what the 4th one was. What's that? Decremental. Good. Then we've got characteristics of an action potential. They're going to be very different. These are all or none. Okay? We either open up all the voltage-gated channels or we don't. Okay? The stimulus was that voltage change. We hit that voltage change, all the channels respond. They all open up. It's non-decremental. There's no weakening no matter how far it goes. So no matter how long that axon is, the action potential at the end is going to be the same as that action potential at the axon helix. Okay? Each time it gets to another cluster of voltage-gated channels, it allows a bunch more sodium to kind of come back in and boost the signal back up until it moves a little bit further. It's irreversible. Once it's started, it can't be stopped. Oops. Yeah, it's a little bit. It's where they're both changes. You know, we're both changing the voltage. There's some very big differences between them. Okay, all or none. Okay, think of it as you're at the fair and you're trying to win somebody one of those big teddy bears by whacking that big mallet and the thing going up and ringing the bell. You either hit it hard enough to ring the bell or you didn't. It doesn't matter how far you got up, they ain't going to give you anything. Okay. In fact, when you ring the bell, they're probably not going to give you anything. You have that big bear, they're going to give you this little thing. Like, well, wait a minute, I wanted that big bear. Like, yeah, you don't win that. That's just decoration. Okay. So these local potentials are you hitting that, and how far up does it go? Okay. If you ring the bell, that leads to your action potential. That's enough to get the action potential that's going to go all the way down. Okay. All those voltage-gated channels will respond to that. It's going to be just as strong as the beginning of the end. Think of that as like dominoes. Okay? If I knock over a domino, the first falls on the second, onto the third, onto the fourth falls on the fifth. The fourth falling on the fifth, if you set them up right, is going to fall just as hard as the 99th one falling on the 100th one, right? Okay. So here is a picture of a graph of an action potential. Okay, and they've actually added the action potential and the local potential. And so our x-axis on the bottom is time, our y-axis is the millivolt change. Okay, they start at minus 70 millivolts because that's our what? Resting membrane potential. Okay, so here inside the cell they're measuring this change. We're getting some stimulation. We're getting a local potential. Okay, sodium's coming in, and it's starting to rise. We're starting to depolarize the inside, making it more positive, more positive, more positive, more positive. Boom. We hit our threshold, minus 55 millivolts. That causes what to happen? All the voltage-gated sodium channels open. Now we let sodium in. Now look at the inside of the cell. It goes from negative 55 millivolts to positive 35 millivolts. Okay? How much of a change in millivolts is that? How much? 90. Yeah, good job. Okay? I like to throw a little math at you guys once in a while. So it's a 90 millivolt change. So look how the local potential was a 15 millivolt change. The action potential was 90 millivolt change. That gives you an idea of the strength of one versus the other. Okay? Cool. Now, we got up to plus 35. And now we start to go negative again. What's happening here? Why would the inside of the cell becoming more negative again? Potassium's leaving. Okay? Potassium's leaving. The inside of the cell. So potassium, let's go back to our start of our resting membrane potential. Potassium was in here. There were two forces. One pushing it out was diffusion. One holding it in was the electromagnetic, because the inside was negative. Now the inside's positive. It doesn't want to be there. All right? Now both forces are pushing it out. And this is our repolarization. It comes down. There's a little bit of an overshoot because the potassium channels were open slightly longer than the sodium channels. Our sodium-potassium pump corrects everything, and we're ready for our next action potential. Does that make sense? Okay. Any questions on that? Just like less than 1/10 of a second, probably. So it's really fast. We're breaking it down so we can kind of get our minds around it all. But yeah, if they were going to do time down here, this might be 1/10 of a second. Okay. So we're feeling this a little bit. We got it. Okay. We're ladies' night in the club. The ladies are inside. The guys are outside in some big line because they got to charge the guys a cover because they're always screwing us over. And as the guys finally come inside, they come rushing in there. You ladies, let us buy you a couple drinks, and then you leave to go somewhere else and find some other suckers to buy you drinks. Because most of you ladies have no idea what the price of a drink is. Never bought one. All right. So, action potentials. A dramatic change in the membrane produced where high density of voltage-gated channels occur. So, produced where high density of voltage-gated channels occur. Where are those two places I've mentioned? The trigger zone and the nodes of Ranvier. Okay? Threshold potential is minus 55 millivolts. So, if we hit our threshold at negative 55 millivolts, all the voltage-gated sodium channels open. So this leg here from 2 on up to 3 are all the voltage-gated sodium channels opening, sodium's rushing in there. Here, at right around 0 millivolts, the sodium channels start to close, and the potassium channels start to open. So things kind of, I wouldn't say slow down, but as they start to close, by plus 35, all the sodium channels have closed, and all the potassium channels have opened, and we start to go back down the other way. So sodium rushes in, depolarizes the cell. Get used to saying that. Depolarization is making the inside more positive. At 0 millivolts, the sodium channels start to close, and the potassium channels start to open. Our graph peaks at about plus 35 millivolts. At this point, we know all the potassium gates are fully open. Potassium is exiting. Okay? Like the ladies from the bar, they're tired of being hit on. And they're no longer opposed by that electrical gradient because the inside's no longer negative. So there's nothing holding them there. Okay? And then eventually we will leave all the potassium, well, not all the potassium. Potassium leaves until we bring it back down in that area of the minus 70 millivolts. We get a little bit of an overshoot we call hyperpolarization. And that was because the potassium gates were open a little bit longer than the sodium gates. Okay? And then our sodium-potassium pumps fix all that for us. Okay? So here's just a picture of that same thing. We're doing a graph along with it. So we're at our resting membrane potential. Sodium gates, potassium gates are more or less closed. We're at our equilibrium. Lots of sodium out here in orange, lots of potassium here in blue. All right? We hit our threshold. They didn't talk about it. We had our local potential. Changed the voltage. Our sodium channel's open. They're just showing 1. Sodium now rushes in. Okay? It's drawn inside to the negative charge, plus it's got diffusion pushing it inside. So both charges push it inside. We make the inside more positive, which we call depolarization. Okay? At about 0, these are going to now start to close. These are going to start to open. That happens until we hit about plus 35, at which point these have already all closed. These are wide open. Potassium now, diffusion pushes it out, and it's being repelled by the positive charge on the inside. It goes out. Okay? And that's our depolarization leg of our graph. So here's our multipolar neuron. Here's where we have our local potentials, sometimes referred to, some books will call them graded potentials. Okay? Graded potential, local potential. If it's enough to hit threshold, we'll open the voltage-gated channels. We'll start our action potential that'll go all the way down to the ends, the synaptic knobs. And they will release their neurotransmitter on whatever they're synapsing on. Okay? Ha! Nice. Do you want to close all tabs? I don't know. That might mess me up. Let's just shrink it out of there. Okay. So what happens specifically at threshold? Of all those statements, which one's the most correct? We all like number 1? All the voltage-gated sodium channels start to open. That's exactly right. All right, so now we get into something known as the refractory period. Refractory period happens just after the action potential. So think of our axon. If this is our action potential and it's moving this way, behind it is a refractory period. The refractory period is the time it takes to kind of reset everything back so that when we open channels again, sodium can come in, potassium can rush out. Okay? If we haven't pumped the sodium back out and the potassium back in, then when we open the channels, we don't get that exchange, right? So we got 2 types of refractory periods. Basically, both of them are a period of resistance to stimulation. We have what's called the absolute refractory period. As long as sodium gates are open, we cannot have another new action potential. So if we have the sodium gates open and sodium's rushing in, and we're at that, say that somewhere between 0 and plus 35 millivolts, it doesn't matter if we hit threshold again, the channels are already open. We can't get another new action potential. Plus the sodium's already rushed in. We can't get much more of that sodium to rush in. It's already kind of flipped its distribution. Does that make sense? So an absolute means as long as we're in this particular state, we can't have another action potential. It's not possible. Okay? We have what's called a relative refractory period. Now, we've gone a little bit further. So now the sodium gates have closed, but the potassium gates are open. And that was that scenario I gave you a few slides ago. If potassium gates are open early, then as sodium comes in, potassium's leaving, right? So the only way to get to that threshold would to be overwhelm it by lots of sodium coming in, and the potassium didn't match it. Does that make sense? So a relative refractory period means that that portion of the axon is resistant to a new action potential. It could still have one, but you need a really, really strong stimulus. You need a lot of sodium. Instead of what would have normally been a 15 millivolt change, you're going to need more than that to get that action potential. Okay? Do those make sense? Okay. So occurs only at a small patch of the membrane at one time and quickly recovers. So I don't want you to think that the whole neuron is in a state of relative refractory period or absolute refractory period. Just a portion of that axon is. So it really limits. There has to be a little bit of a space between one action potential going down the axon and the one right behind it. Okay. This isn't a good graph. Let's ignore that. Okay. Do we have any questions on the refractory period? Absolute, relative. Okay. So now we're going to talk about this impulse propagation in unmyelinated fibers. They like the word propagation. You'll still see books and things talk about conduction. But conduction truly, if you were talking about conducting, it would say the sodium that ran in here rushed all the way down to the end, and that was our action potential. It was conducted from the beginning to the end. And we know it's not this sodium molecule. This sodium molecule led to another sodium molecule to another. Now, they're all identical, but it's not the exact same one, so they use propagation is the word. So we hit our threshold voltage, reaches that in the trigger zone, and this begins our impulse. And this impulse that's being propagated is basically a chain reaction. Okay? So we've got an axon. It goes from this wall to that wall. Okay? And in this little patch right here, we hit our threshold. We opened up all the voltage-gated sodium channels. Sodium rushes in. Okay? Sodium comes in the membrane, and they're not very deep. It comes in and has to go left or right. It goes down the axon a little bit. That change in charge right there from the sodium with its plus charge made the inside positive enough that we opened up all the voltage-gated channels right here. That rushed in, caused all the voltage-gated channels to open up. Sodium rushed in, moved down, and it goes down, 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 down the axon. Okay, like a domino. Okay? That's in an unmyelinated fiber. A sequential opening of voltage-gated sodium channels all the way down, one after another, down the axon. Okay? Go all the way to the end. And the way the axon's set up, the amount of sodium that comes in here is equal to the amount that comes in here. It's non-decremental. It doesn't get any weaker. At the end, it's the same strength. But without the myelinization, it's relatively slow, at least compared to, say, the type A fibers. Okay. So here's an axon. They've taken a little section right here, blown it up. And the green part, this is kind of the axon at rest. The inside's negative, the outside's positive. Okay? Well, we've started to get some sort of action potential where the inside becomes positive. That leads to this one becoming positive because what's happening here? Sodium's rushing in, right? Triggers sodium to rush in here, make this positive. And that's what it's doing. It's moving down the axon. Behind it is that refractory period. Okay? It's not specifying whether it's the absolute or the relative. Here, if we wanted to split hairs, we could say this is probably the absolute. Here's the relative. And by now, it's been reset. The sodium or potassium pumps have reset that unequal distribution of electrolytes, and we're ready for another one. So you could have an action potential here, and then here. You just have to have a little space between them. Okay? Does that make sense? Now, saltatory conduction is the term they use for propagation of the impulse with a myelinated axon. And it's real simple how this works. Okay? So, pretend you don't see the myelin sheaths. Let's pretend they're not there. We got a large number of voltage-gated channels. Sodium rushes in. And if we didn't have this myelinated, let's say it would rush in here and do the same thing again. Open up another sodium-gated channel and rush in here. Move down here. Open up another one. Another one. Another one. Another one. Well, each one of those takes time. What the myelin sheath does is it insulates it so that it rushes in and there's no, it can't be pumped back out. So while it's in this portion wrapped up by the Schwann cell, the sodium's going to rush all the way in from here to there. And that movement of sodium is really fast. The thing that's a little bit slow is the channels opening up. So now, instead from going from here to here, instead of opening up, say, 15 channels, it opens one here and opens the second one there. Okay? And the sodium rushing in is very fast. So it's like the impulse jumps from here to there. Okay? So my analogy for that would be as if we went out on the football field and we had some sort of ball. Okay? And we had to get the ball from one end to the next. And I lined up one group of you 5 yards apart. You had to catch the ball and turn it to toss it to the person 5 yards away from you. And we had another team, considering you could all catch and throw, 20 yards apart. Which one's going to get the ball to the other end zone the fastest? Yeah, the one that's 20 yards apart. Because the fast part's throwing the ball, right? Catching it, turning to throw it, that's the slow part. That's the channels opening up. So if I space out the distance that one impulse goes before it has to open up channels along the way, I've just sped up the process, right? Okay? So go back, because I see some eyes that are like, So instead, if this wasn't here, if this was unmyelinated, this sodium rushing in would move to here and open up another channel, which takes time, and have sodium rush in to here. Open up another channel, sodium would rush into here. Open up another channel, sodium would rush into here. See how that's a little bit slower? Each time we open channels, it's slower. This, sodium rushes in, goes all the way down here, opens up the voltage-gated sodium channels, because we said that's where we have high concentrations of them, as are in those nodes of Ranvier. Okay? So voltage-gated channels are needed for the action potential. They're located in the nodes of Ranvier. So if we can limit the sodium being able to escape once it gets in there, and it moves very quickly, then that impulse is sped up as the sodium moves that greater distance between the nodes of Ranvier where the axon's actually myelinated. Okay. Ranvier, yep, that's the space. Okay. So here, notice we're not worried about any voltage changes in there. That's the insulated portion. So the action potential goes from here to here to here. It goes much faster. It's like it jumps from one to the other. Okay? Where the unmyelinated would have to be opening up channels in this thing, which would slow it down. All right, let's see if we got a video that works. Yeah, that's not doing much. All right, well. Hey, come on back. Win. All right, so now we're at a synapse, which we've seen before. We're familiar with synapses. We saw a neuron synapse on skeletal muscle, right? Neurons can synapse on each other. One synaptic knob could synapse on a dendrite or a soma or cell body of another neuron. It's the same mechanism. They release neurotransmitters. Okay? So when we have a neuron synapsing on a neuron, they get really technical. They name the first neuron, and the second neuron, the second neuron. Do you need me to repeat that? Be a first-order neuron or a second-order neuron tells you where they are in the synapse, the one first or the one second. Sometimes they'll call one the presynaptic neuron and one the postsynaptic neuron. The presynaptic neuron synapses on the postsynaptic neuron, the synapse being the thing in the middle. Okay? So the first neuron is the presynaptic neuron, the second one's the postsynaptic neuron. And they can describe these on what the presynaptic neuron synapses on. We can say it's axodendritic, meaning the first neuron synapsed on a dendrite. It can be axosomatic, meaning the first neuron synaptic knob synapsed on the soma or cell body. And we can even have where the first neuron synapsed on the other one's axon. Not nearly as common. Okay? The other thing that's very important is to know the synapse number on the postsynaptic cell varies. For us, we're going to be concerned that there's many presynaptic neurons synapsing on any one postsynaptic neuron. Okay? Lots of pre's on one post. Okay? Each one of those neurons that synapse on it might fire, might not fire during any given situation. They're all telling the postsynaptic neuron something. They may be telling it to fire. They may be telling it not to fire. They may not be voting at all. But the more presynaptic neurons that synapse on a post, the more input that postsynaptic neuron gets, right? Just like if you were going to make a decision, the more people you get input from, the more you know what's going on. Okay? If I wanted to know something about campus life here at Phoenix College, if I asked 2 of you, I probably wouldn't get as clear a picture as if I asked 22 of you, right? And these chemical synapses allow for there to be integration of the information. Okay? Basically saying, if you have lots of presynapsing on a post, it's like each presynaptic neuron gets a vote. Okay? And they give that input to the postsynaptic neuron, and depending on how that all comes out, the postsynaptic neuron fires or it doesn't fire. Okay? You're in your relationship with whoever, you got all your friends, you're like, yeah, maybe I should break up with him or not or whatever, and they all give you some advice. Okay? You take all that advice, probably don't listen to any of them and do the wrong thing. Okay? Well, the neurons actually listen to the other neurons, and so they do what they're told. Okay? All right, we'll stop there for the time being.